From the JAMA Network, this is JAMA Dermatology Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Dermatology.
Hello and welcome to this author interview from the JAMA Network. This is Dr. Adamson, the web editor for JAMA Dermatology. Dermatophytosis is a common contagious superficial skin, hair, or nail infection caused by dermatophyte fungi, which are most commonly members of the genus Trichophyton. These infections are typically easily treated with antifungal medication, such as terbinafine. However, in the past decade, dermatophytes with resistance to typical antifungal dosing and duration have begun to develop around the world, leading to growing public health concerns. In a new study in JAMA Dermatology, authors present a case series of 11 patients in New York City infected with Trichophyton indotineae, a recently described dermatophyte that causes extensive pruritic plaques which do not resolve with typical antifungal treatment regimens. Dr. Sudha Chaturvedi is the director of the Wadsworth Center Mycology Laboratory within the New York State Department of Health in Albany, New York. She is also an associate professor in the Department of Biomedical Science within the School of Public Health at the University of Albany in New York. She is the senior and corresponding author of this new study and is here to discuss this work. Welcome to the podcast, Dr. Chaturvedi.
Thank you.
So it's not typical that we have mycologists on the podcast. So do you mind telling our listeners a little bit about your role? Because it seems like it's split in academic appointment as well as someone who works with the New York state government.
Yes, so Wadsworth Center is a public health lab, and I work here as a research scientist. And also we are affiliated with the University of Albany, through which I have a appointment as associate professor in biomedical sciences.
And do you have a special interest in fungi as something that you study either on a molecular level or from a public health level?
Absolutely, sure. I've been working with fungal pathogens for last 24 years in Wadsworth Center, and I have been director of the mycology laboratory for more than almost 10 years. And we do a lot of testing. As part of public health lab, we have to do surveillance activities just to see which fungi are evolving, whether they are developing resistance, where they are in the United States, where they are in New York. And we also have grant from CDC now, and we are one of the AR lab network for the Northeast, where we are mostly involved with Candida auris. So we do a lot of surveillance activities, but apart from that, all the labs here in Wadsworth Center, they also have a lot of interest in developing new testing, where the whole idea is how fast we can reach from bench to bed, in the sense that, we want to take care of patient care and how a lab can provide that information to clinicians. So we develop a lot of testing in the laboratory, which is molecular in nature to most part. But we also have a lot of classical testing also we do in the laboratory.
And for this specific study, you were looking at infections related to Trichophyton indotineae. How did you get interested in this specific pathogen?
So we also do a lot of reference testing in the laboratory, where pathogens which are difficult to identify by any microbiology laboratory or clinical laboratory comes to us. And in this process, we also interact with a lot of physicians, technicians, lab directors. And this study came to our attention, and Dr. Avron Kaplan, he is a dermatologist in New York City, one of the hospitals. He actually came up with, suspicion that this could be Trichophyton indotineae, and he sent specimens to us, and we have done the molecular testing, which we anyway run in the laboratory for other fungal pathogens also. And It turned out to be Trichophyton indotineae. Those were the very first two cases from New York City, and we published that work in CDC journal called MMWR. And then, the interaction begin between the lab and the clinicians, and most important one is definitely Avron Kaplan, and then also CDC was involved. And I should say Dr. Jeremy Gold was another epidemiologist from the CDC who was actively involved in trying to understand this pathogen, how it is disseminating in different parts of United States. But we have so far only focused on working in the patient from New York because we are public health lab for New York. So that was one of the reasons. And also the physicians who are involved are from New York.
And in this particular case series, there were 11 patients that ended up having this diagnosis of Trichophyton indotineae. And they presented with these really itchy plaques on different parts of their body, or their trunk, extremities. And these lesions were really difficult to treat. All the patients had at least some kind of course of topical medicine. Some of these patients also failed oral medications as well that we typically use with the treatment of dermatophytosis, like fluconazole, griseofulvin, terbinafine. But ultimately, itraconazole treatment was successful in all of these patients. Is this the typical presentation that these patients have? It's just really difficult to treat fungal infections. What would you tell dermatologists that may be encountering this kind of diagnosis?
These cases don't look very different from anything else. What happens is, I think that's why it's very important that we should publish all this work into reputed journals like yours. And also, we are going to send an advisory to all physician and clinical labs about the emergence of Trichophyton indotineae. That's one of the role of public health lab like Wadsworth Center, so that clinicians know that Trichophyton indotineae has reached the United States and is also infecting people other than other dermatophytes. So as far as the diagnosis is concerned, currently we don't have any simple methods where a simple clinical laboratory can identify this as T. indotineae. They generally identify this as either mantegrophyte or interdigitally. What we tell them that please send anything either mantegrophyte or interdigitally to us, and we run a molecular test, and that is able to identify that to species level indotineae. So definitely at this point of time, the molecular test is the only way to speciate this particular fungal pathogen. And why this is making so much news, if you want me to continue on that, is because it's quite resistant to first line of drug called terbinafine.
Mm-hmm.
And that was is happening that, that's the choice of drug for dermatologists. They use it and they find that patient is not doing well. So that kind of gives them suspicion. And that's the way we started getting more and more now this particular species called indotineae in our laboratory.
Do you think that indotineae is underdiagnosed? Because it sounds like the typical lab does not necessarily have the ability to speciate. So for our listeners that may not be in New York, where could they send their specimens if this is something that they are concerned about as the diagnosis?
Yeah, you're absolutely right that it is definitely, we think it is underdiagnosed because unless you have a real sense of this emerged in United States, even physicians probably are not thinking about indotineae. And yes, there are only very few labs currently which can identify this particular fungus to species level. We are one of them. CDC can do that. There are several reference labs, they can do that. And also there are several commercial labs. They have the way to identify this fungus to species level. But in general, any micro or clinical laboratory who do not have a molecular way of identifying fungi have the capability to ID this fungus.
And so stepping back and thinking about this fungus, it used to be called Trichophyton mentogrophyte genotype 8. So this may be one of the reasons why traditional laboratories may not be able to identify it. And do you think one of the reasons why it's a resistant organism is because of the use of combination drugs that have both steroids and antifungals in them that are commonly prescribed in the United States? Is that one hypothesis as to why we're getting these resistant organisms? Similarly with antibiotics, is this a case where we're just using too much and there's not as many calls about the overuse of antifungals as there has been about antibiotics?
So first of all, I would like to say that Trichophyton mentogrophyte genotype 8, it was previously known as that, but that genotype is also not easy for any clinical labs to identify because there are several genotypes within the species complex. And unless and until you run a molecular assay, you cannot differentiate one genotype from another genotype. So for that reason, I don't think so any clinical lab right now who don't have any molecular technology can identify this fungus even to genotype level. And in terms of your next question, what made this as emerging resistant pathogens? So this all started most likely in India, we think, because the drugs are very easily available. People have access to drugs, especially the topical ones. And also corticosteroids are also easily available. So there is misuse of drugs, topical drugs. Additionally, even when patients have infection, the dermatophytes, they are not causing any death, right? They are only causing a lot of itchiness, uncomfortable. You're uncomfortable. It's a morbidity, yeah, if you want to use that word. It's not mortality, but morbidity. So patients, if they get little better, they start stopping the treatment. And no patient is in the hospitals, so the physicians cannot see them every day whether they are taking their medication or not. So it's a combination of factors, misuse of both topical as well as, I will say, the oral drugs. So that led to development of resistance in this particular pathogen. And this is very contagious pathogen. It also makes tons of spores, which is very easy for it to disseminate from one place to another place. So having this affinity for humans, highly sporulatory nature of the pathogen, and also misuse of drugs, it's like combination of all these things led to emergence of this particular species, I will say. Trichophyton, now we call it Trichophyton indotineae. The name has been given, I guess, in 2020. It's no longer called Trichophyton mentogrophytes genotype 8.
And do you think by having a new name, it will help in being able to be alerted about this as a potential diagnosis? And perhaps we can then have patients treated with greater quickness, because a lot of the patients that are in this case series had tremendous delays in their diagnosis, and then ultimately, as a result, the definitive treatment, which for most of them was itraconazole.
Yeah, I think it's not going to make big difference. Name change happens in mycology as well as in other fields based on when that genotype shows number of characteristics feature which is away from other genotypes, like is highly virulent, has affinity for humans, it has developed drug resistance. Those were some of the criteria, and it came mostly from India. So those were most of the criteria for speciating this into indotineae. And the awareness is very important, not only among laboratorians, but also among physicians, among clinical labs, among lab technicians, that this species is now emerged. It's no longer a foreign species. It's very much here in the United States. I think we need more communication, we need more publications, we need
More awareness.
all kinds of informations to provide more awareness, absolutely. So then the whole healthcare system has an idea that this fungus is very much here. And then once physicians know, I think they are going to treat with the right kind of drugs. And the testing is also very important in terms of antifungal susceptibility testing, which is definitely, I will say, is very rare and not every lab does that. So it has to come to either reference lab or public health lab or commercial labs. And they are not everywhere, right?
Yeah.
Even from New York City, everything is coming to Albany. That also delays the whole process.
One more thing I would also say that was very interesting about this study is the patients, by and large, were immunocompetent patients that had extensive disease. So this is not necessarily a pathogen that we see, at least for now, that's disproportionately in people that are at higher risk from an immunologic standpoint.
That is true.
Is there anything that surprised you about this case series that you'd want to tell the audience to keep in mind?
I was thinking that probably this has all came from India, but then we have done the whole genome sequencing, and we found out this our cluster of 11 patients looks like they all originated from Bangladesh. So that was a little different than what I was anticipating. And other than that, we also have done modeling, which I felt was needed because we knew that there is a mutation in this gene called squalene epoxidase, and which results in resistance. But we didn't know exactly where it is situated and how that mutation is responsible for binding of terbinafine. So having that information is worthwhile to discuss. And that's why we put in everything together in this publication, and hopefully everyone will benefit out of what we are trying to say here in this case series, I will say.
My name is Dr. Adamson, and I've been speaking with Dr. Sudha Chaturvedi about this new emerging pathogen called Trichophyton indotineae, which we need to learn more about, not only as dermatologists, but as clinicians. And it is also an important reminder about the stewardship of not only antibiotics, but in this case, antifungal medication. You can find a link to this paper in the episode's description. This episode was produced by Daniel Musisi at the JAMA Network. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com. Thank you for listening. Catch you on the next episode of the JAMA Dermatology Podcast.